Alright, in this video we're going to talk about the bonds that make up macromolecules. Each one of them has a unique type of monomer and a polymer name. So we want to focus on the macromolecules that we've talked about here, but how do we build those polymers and what are the names of the bonds? Each one of these has a specific name. And so, brought to you by Curious Marineland. So when you actually see this right here, and actually that, and I do have a video for lipids, so I'm going to go in order and we're going to talk about what exactly does it mean to actually add a molecule specifically for carbohydrates, lipids, proteins, nucleic acids. And here are the bond names that you're going to hear in this video. So what I've done is four short videos. I've combined them. I'm going to go in this order, and we're basically going to talk about how these are made. I'm going to use models, and I'm also going to use some pictures. So this is just my intro to that. So stay tuned, and next we're going to talk about carbohydrates, all right, and then lipids, proteins, nucleic acids. So what do you see? I see 1, 2, 3, 4, 5, 6 carbons. I also see 1, 2, 3, 4, 5, 6 carbons. So I'm going to arrange these carbons so that the fourth one here is going to interact with the first one here. And this, when it's combined, will be a disaccharide, which is a carbohydrate. So if you notice, look at the model. Here's the linear form of glucose. Here's the cyclic one. So you see 1, 2, 3, 4, 5, and 6. So 4 of this one will interact with the 1 of another one. So let's look at it. Here's glucose and fructose that makes sucrose. Here's glucose and another glucose. That's what I'm going to demonstrate. So you'll see 1, 2, 3, 4. So here's 4 right here. And here's 1. And this right here is going to be a bond, AC. And this is the bond that connects one carbohydrate, a monosaccharide, to another. And this is called a glycosidic linkage. Glycosidic linkage. So I have this little purple bond just to show what's going to happen. Typical dehydration synthesis. So dehydration synthesis. So when these molecules combine, some water has to come out. So let's do this. So here's the OH right here. And then I'm going to take off here. You can always show water being produced, and you'll see this in all the videos I do for this dehydration synthesis with the molecules. So here it is. This is a disaccharide. In particular, it's actually maltose, which is a sugar that you see in malt sugars and stuff like that. So that's pretty much the first one. I'm going to do a video for lipids and proteins. For nucleic acids, we're going to do like more of the PowerPoints, but I'll also show you what the model actually looks like. So that's the first of a couple, and the whole idea is build a molecule or break it, biosynthesis or digestion in all essence. All right, next one video is coming. Now let's talk about lipids. Now a lipid, if you look at this model, and you'll see, I'm going to go back to the PowerPoint in a second, it's actually not complete. So what I want you to see is, this is a 1, 2, 3 carbon chain called glycerol. Now it turns out each one of these glycerols has a fatty acid attached. Fatty acid attached. Now if you call that 1, 2, 3, I haven't added 1 to 1. I'm going to do that in a second. But so what is it? A triglyceride, 1 glycerol. And so if you see right over here, this is the glycerol. And now watch what happens. You add in a fatty acid. This did it to the middle. Add in another one. And there you go. This is actually called a ester linkage. Carbon, oxygen, carbon, that's an ester linkage. So I'm going to talk about all the macromolecules, how the actual bond is created. And this is a series where I'm using the models. So we'll do all of them. We'll do carbohydrates. Now we're doing lipids. So let's go ahead and just make this ester linkage. So let's say, so here we have, it's a classic dehydration synthesis. So what I have to do is, as you see here, I'm going to take, there's a hydrogen, and there's a OH. Here is your water. Water comes out, and here, as you look at the purple bond, is your ester linkage. Now I actually have a triglyceride. I have a glycerol, and coming off each glycerol, carbon 1, here's a fatty acid chain, here's a fatty acid chain, and here is a fatty acid chain. Actually, we have a unsaturated here because it's a double bond. This one right here, the fatty acid chain is single. So that's saturated, saturated, unsaturated. So that's a short little look at lipids. So we're going to move on to our next model, and that will be proteins. So take a look at the models. This is two amino acids. They combine to make proteins. So let's look at the four parts, and then we'll combine these. So I have two amino acids, and we're going to arrange it so that we can combine these two. It's classic dehydration synthesis, but let's identify the parts. You got a central carbon. So here's your central carbon right here. And then you're going to have a simple hydrogen. Then you have an NH group called amine, and you have the carboxylic acid. That's where the name amino acid comes from. Okay. And then you got this R group. Now this R could be as simple as the glycine amino acid that has just another hydrogen, or it could be something as big as this molecule right here. All right. As you look at these different ones, this picture is showing they didn't put the actual formula, they just put the name of the different amino acids. So let's show a peptide bond being formed. There are 20 amino acids, and again, this is part of my series on identifying the names of the bonds and using models to do so. So let's go ahead and adjust. What you have to do is arrange it like this. You need to have, here's an amine, here's the acid. Here's the amine, here's the acid. So they combine here. This should be your classic dehydration synthesis. When this peptide bond is formed, you should be able to account for everything. So let's do that. What we have, so here is, coming off the carboxylic acid is the OH. Coming off the amine is the H. Here's your water, dehydration synthesis. The purple bond, like I've used before, is your peptide bond. This is a dipeptide. Take another one and put it here, and another, and another, and another, and you now have a polypeptide chain, like so.